Alright, we're going to talk about advances in genetics, and this is going to go on page 23 in your science log. So right now you can update your table of contents and write page 23, advances in genetics, and today's date, which is February 6th. This is going to be talking about things like cloning and selective breeding and genetic engineering. So on page 23, selective breeding. This is when we select or choose organisms with desired traits and crossing them, hoping to get those traits in our offspring. So people have been doing this for thousands of years, especially in agriculture, when they noticed something that tastes good and they had another plant that grew large fruit. They're going to cross the large fruit with the tasting good fruit and hopefully get a large tasty fruit. So they're doing this lots of times in the past, and with animals, if they had a fast racehorse and another fast racehorse, they would hope by breeding them they would get an even faster racehorse. So the thing to remember about selective breeding is that people are doing the choosing. They are choosing which organisms to breed. There are two types of selective breeding. The first type is called inbreeding. This is when we cross organisms with similar traits. So in the examples I talked about before, the horse race, the horses would be an example of inbreeding, trying to get faster and faster horses, so we keep crossing them. Another example is when you see those contests in October to get those giant pumpkins. They started off with pretty big pumpkins and they kept breeding them and breeding them and breeding them until they got really large pumpkins. And they used other things as well, probably fertilizers and different things like that, but they're trying to get the genes that are going to create something extremely large, and that's how they do some of those agricultural things that they make. So that would be having the same traits or similar traits. That's called inbreeding. Some problems that can happen is we can lead to genetic disorders because we are not mixing up our gene pool. We keep trying to get the same genes and the same type of organisms over and over, so if there's any kind of recessive disorders, those are going to start popping up more frequently. So this happens a lot in pedigree type dogs and cats that you would see on TV in the dog shows. They don't have a very large gene pool, and we've talked about this before, and we can get quite a few problems because of that. The second type of selective breeding is called hybridization. This is when we get two different traits and we try and put them together. So a tangelo, if you've ever had a tangelo, is a tangerine crossed with an orange. So that's a hybrid fruit. Okay, there's lots of different kinds of hybrid fruits out there and vegetables that they cross. And you probably are thinking of some animals, and we'll talk about those in a little while, that you might have seen before where they cross two different animals that aren't the same species. And we saw, some of you have seen this Bill Nye, where they cross a donkey and a horse and they get a mule, or a mule and a horse and they get a donkey, I forget. But that's one example. Okay, another example is farmers crossing corn that have lots of kernels with corn that is resistant to disease. So they're hoping that they're going to get lots of kernels that is resistant to disease. They're hoping they get those two traits. Does it always work? No. Sometimes it doesn't work. Sometimes they might get traits they don't want showing up. But they keep trying until they get something close to what they're looking for. Alright, another advance in genetics is called cloning. We've talked about this already. It's making an exact copy of an organism. So there are some things in nature that can just clone themselves without any help from humans. And bacteria can split themselves in half, and basically they're making a clone because they have identical DNA. A sea star, when its arm is cut off, and if it's cut in the right spot, it can grow into another sea star. There's some other organisms that go through something called budding, like a hydra will bud and it'll create another hydra, and a sponge will do that, another exact copy, and that's kind of like natural cloning. The DNA will be the same, but the organism might be different depending on the environmental factors. So if we have a clone of something but it's not fed as much, or it's not in the same environment as another as its clone, it might turn out a little differently. Types of cloning, we have two different types. One is cutting the stem from certain plants and then planting that new part, and that will give it a cutting. That's called a cutting. It'll grow into exact same plant. Some plants do this on their own. They don't even need any help from us. And then we've talked about this. We saw the video on this, taking the nucleus out of an animal cell and putting it into another cell and then trying to grow the new organism. So number 2 was how we saw Dolly was made. They took the nucleus out of a cell, they put a different nucleus into the cell, they had that egg cell grow in a different sheep. Remember, it took 3 sheeps to do that. For the first kind, the cutting the stem, that happens a lot. Some plants actually send out little runners that make a new plant. Strawberry plants will do that. They'll send out a little runner, and then it'll grow another strawberry plant maybe a couple feet away from the original strawberry plant, and they have identical DNA. Okay, so there's kind of a natural way of cloning, and then there's the scientific way where we have to manipulate it to get a clone. Okay, genetic engineering is when you take DNA from one organism and you put it into another organism's DNA. So this could happen by scientists taking a certain gene out. Let's say they wanted the gene for bioluminescence. We talked about this. And you take the gene for bioluminescence out of a bioluminescent organism and put it into a different organism, and then that organism would glow. So we call that splicing the DNA. When we cut it apart, we're splicing it. They use enzymes to do that. And we don't really need to get into exactly how that happens, but enzymes will cut it at certain spots. It'll read the DNA, it'll know when to start cutting, and then they'll get the piece that they want. So some of the things that are genetically engineered are crops. A lot of the crops today are genetically engineered to survive pesticides or herbicides. They will spray heavily with things that will kill weeds or kill pests, and they don't want it to kill the plant. So they genetically engineer the plant to be able to survive these things, these harsh chemicals, so that they don't have to worry about how much they spray and how much they put on the plants. Also, livestock are genetically modified. They want to have the biggest cow or the biggest chicken with the most breast meat, or the biggest animals or birds, so that they can grow as quickly as possible and they can get as much meat out of them as they can, because that's why they make more money. These are called genetically modified foods, or GMO, genetically modified organisms. So you may have seen that some foods in the grocery store are now being labeled as non-GMO, which means they haven't had any genetic engineering, and they're more what they call natural types of foods. So some people, you don't have to write this part down, but some people have concerns about GMOs, and I want you to just think a minute, talk to your neighbor. What do you think they might be worried about? If they don't want to buy food that has been genetically modified, why wouldn't they want to do that? Talk to each other about it for a minute. All right, and then what I want you to do tonight is go home and ask somebody what they think about genetically modified organisms, and if they don't know what that is, you can tell them, and see if you can get a little conversation going and find out what other people think about it. Okay? All right, now here's what you're going to be doing today. You're going to be creating some animal hybrids. So I'm going to show you, talk about some and tell you about them, and then I'll give you the directions of what you're going to be doing. In captivity, biologists can breed certain animals that are in the same family or the same genus, and they try and get desired traits from both of those animals. So you have seen some of these, or you may have seen some of these before. An example would be a zebroid, which is a zebra crossed with a horse. So they're hoping to get the speed, a faster version of a horse or a zebra. That's what they're trying to do. And then in this picture we have a zonkey, which is a zebra crossed with a donkey. Donkeys are very strong, and they're pack animals, and so they're trying to get a stronger kind of pack animal. All right, and they're both horses. It's not like they're both in kind of like the horse family, not necessarily horses, but in the horse family. They're not going to take a cat and breed it with a fish. It's not going to work. So they have to be in the same family or genus. And another animal hybrid that I haven't seen this one before. It's a camel crossed with a llama. Okay. Here's another one. It's a pheasant crossed with an Amherst. It's a different kind of bird. So it's kind of interesting. And then you guys have all seen this. If you've seen Napoleon Dynamite, a liger is a tiger and a lion. And like we talked about before, a lot of times we can get some problems when we're not, when we're messing around with this. And ligers, I know they don't live very long. They have a lot of problems with their circulatory system. And so it's really not the best of both worlds when you start doing things like this. It doesn't always work out great. But what you're going to do is you're going to create an animal hybrid using the paper that you're going to get in a minute, and it's going to give you a bunch of different boxes, and you can choose from those boxes. You have to choose the things that you're going to mix together from the same box. So if you wanted to do a wolf crossed with a coyote, you could do that, but you could not cross a wolf with a bobcat because those are two different boxes or groups. Okay, so you have to pick from the same group. And then at the bottom of the paper, there's a paragraph that you're going to write on your paper. You're not going to write on the direction sheet. You're going to write on your paper, I am crossing a blank and a blank. I want, sharp teeth from the tiger, and I want, brown fur from the lion so it can blend in and have really sharp teeth. And you tell me what you want to have happen. Okay? We're not going to do the inbreeding part. Okay, so just don't worry about that sentence at the very bottom. And then you have to draw and color your picture. Draw and color your picture and give it a name. Okay? And then you're going to be turning that in. If you don't finish it at the end of class, you will be turning in on Monday. All right. Hope you have a good time making your animal hybrids, and I will hang some of them up outside when you turn them in.